start the recording as well. So first of all, welcome to this first lockdown lecture of AUUC, and hopefully it will be one of a couple to follow, so that we can all help each other to get through the lockdown period. So for me, I will be the first one to do the first presentation, and it will be about breathing gas physiology. And I hope to make it as interactive as possible. So first a little bit about me. I'm Xavier Vrijdag. I am a PhD student at the University of Auckland, looking at the effects of gas narcosis in diving, where I use EEG, so the electrical signals of the brain, in order to make a monitoring method to hopefully get a better understanding of how narcosis is working and how each of the different gases that some of them we'll discuss tonight as well, how they are affecting the brain. As a diver, I started diving at a young age because both of my parents were divers, and from the beginning I got really interested in diving. So all the way through high school and university, everywhere possible, I chose projects towards diving and diving medicine, and well, that in effect is why I also chose my current PhD topic. In diving, I am diving in caves, in deeper wrecks, using trimix mixtures and decompression gases. So a lot of the knowledge I will be sharing with you guys will be quite well known, possibly to some of you who are technical divers, although I will try to make it as basic as possible, so I hopefully can accommodate all of you. So in this presentation, I will cover the effects of nitrogen, and especially nitrogen narcosis. And I also will go into the effects of oxygen, both hypoxia and hyperoxia. And the last gas I would like to discuss tonight is carbon dioxide and the toxic effect when we get hypercapnia. So first of all, nitrogen. In a more broader sense, we use the term inert gas narcosis, because there are a couple of gases that might have a narcotic effect on our brain, and nitrogen is one of them. So what we experience if we go deeper is that we get a similar effect as alcohol, where our brain doesn't perform that well anymore. And there are a couple of theories, and over time they have been, most of them have been proven wrong. So the ones that we know in the diving industry that are most commonly mentioned are the Meyer-Overton rule or theory, and three others: Castell's metabolic theory, the Klatterath theory, and the iceberg theory. The later three are less mentioned, but the Meyer-Overton theory is used quite a lot, and especially you might be familiar with the solubility coefficients in olive oil. Because Meyer and Overton found both that the narcotic potency of various gases is linear with the solubility of that gas in olive oil. And they also did some further research and found that they formulated the idea that the gas molecules would go into the cell membrane and widen up the cell membrane, and hence would jeopardize the conductivity of electrical signals in the neurons, and hence in that way would cause their narcotic effect. Later on, this was proven wrong, so that theory is not used anymore, although even it's still a common thing to be used in the diving industry. But in anesthesia, they found that it's not the cell membrane that's actually affected, but the but certain receptors on the cell membrane that follow a certain pathway to get signals from one cell to the other, and the narcotic gas molecules are prohibiting that cell conductivity, and hence in that way they cause their narcotic effect. In diving, we think that some divers are more susceptible to inert gas narcosis than others, and that you can somehow be immune for it. Several studies have shown that that's actually not the case, and that all divers are affected equally. It's just that if you've done it a couple of times, you get more used to the effects of inert gas narcosis, and hence you can cope with it better. But it doesn't mean that the effect is not there anymore. And the coping effect works best in an environment where you are doing stuff that you are pretty familiar with. So your brain doesn't need that much capacity in order to execute the things you want to do. But if you're suddenly faced with a novel environment or a novel thing you have to do what you haven't practiced before, then it becomes really hard to actually perform the task that you need to do. So let's go to the symptoms of inert gas narcosis. As I mentioned before, they are similar to the effects of alcohol intoxication, although there are some differences there as well, small ones, but mostly alcohol works differently on the brain than nitrogen does, although the end effect is relatively similar. Inert gas narcosis becomes more pronounced with an increase in depth, and that's mostly because there's an increased partial pressure of inert gases, mostly nitrogen. And hence, if we ascend, we have a decreasing partial pressure of nitrogen, and hence a decreasing effect of gas narcosis. There is a onset time at depth. The first couple of minutes, it takes a little bit of time for the nitrogen to get absorbed into your body, into your brain tissue. So it takes a couple of minutes before there's a full onset of the narcotic effects. Some divers, especially those that go deeper, and participants in my study can probably relate to that as well, that the first couple of minutes kind of woo, there's that onset effect. And after the first couple of minutes, you kind of get used to the effect and you can work with it better. There are big intra and inter-individual differences, which means that on the same doing the same dive a couple of times on separate days might mean that one day you're more affected than other days, where there's also a differences between divers, as I discussed before. So the symptoms depend on depth, as I discussed before. And so in the shallow, it are mild effects, and really, if you monitor yourself closely, you can find those effects already somewhere between 20 and 30 meters. However, the deeper you go, the more onset of symptoms you get, and the more um, your brain gets affected, and the less normal effects you can do. I will come to that question in a moment, Ryan. So the symptoms get progressively worse, but the biggest problem with these symptoms is that they alter your capability of monitoring your environment to make sure you stay safe, and if there's something wrong, to make the right decision. So hence you can get into a spiral where you make one stupid mistake, you don't recognize the effects of that, and you get in a downward spiral where at some point um, you make the wrong mistake, and you can actually kill yourself. So the treatment is relatively simple. Ascend and make sure that in your planning phase you select the right gas mixture, which I will come on to later on as well. And make sure that you take the right gas for the right depth. If you want to ascend, if you notice that on that day you are affected more than normal, ascend, make a shallower dive, or stop the dive and ascend all the way to the surface. Symptoms should disappear quickly, although there might be some residual symptoms like memory impairment, and that's mostly pronounced in a good story that a couple of divers have told me separate of each other, is that in the UK there's quite a lot of diving happening on wrecks around the islands, and quite a lot of them are in the depth range between 30 and 50 meters, and quite a lot of them have been diving back in the 80s and the 90s, those dives on air. And some of them made at some point a switch to diving those same wrecks on trimix, using helium to reduce the amount of inert gas. And although they've been diving numerous times on those same wrecks, once they did the same dive again on trimix, they noticed all the small little details of that wreck that they couldn't remember from previous dives. Same effect is that with trimix, the dive was much brighter. There was much more light. It didn't feel as dark as it would have been diving those same dives using air. So Ryan, your question is that do symptoms depend on the gas, or do all inert gases produce the same symptoms? So in the end, given that most of those inert gases affect the GABA receptor on your neurons, they will exert a similar pathway of a similar logic of symptoms. It might just be that their potency might mean that there are different partial pressures needed in order to get the same effect. There are a couple of exceptions, luckily not gases used in the diving industry, but for instance, laughing gas, nitrous oxide, and ketamine are anesthetics used which don't affect the GABA receptors, but the NMDA receptors, and hence have a slightly different pathway on how they affect the brain. So that is slightly different, but the low dose effect of these two anesthetics are in the end quite similar to the effects of, for instance, nitrogen in diving. So the next gas that I would like to discuss with you all is oxygen. Oxygen is a necessity of life. Without it, we can't survive that long. It is also a drug used in hospitals, in emergency departments, in the ambulance, but also in home care. And as divers, we use it as a drug to treat decompression sickness, and we even might increase the pressure while breathing 100% oxygen in a hyperbaric chamber. However, it's also a poison. So first of all, I would like to discuss hypoxia, the shortage of oxygen. And this can happen because you are breathing from a gas supply that has an inadequate amount of oxygen. This can be due to a faulty rebreather or a gas mixture that didn't have the correct amount of oxygen needed. But secondly, we need to get that oxygen into our body and into our cells in order to sustain the life of those cells. So even along that path, there can be problems that can cause hypoxic effects, like an inadequate ventilation. So if you, although you might be breathing a gas that has enough oxygen in it, if you don't breathe enough, you will get hypoxic, like a free diver. Then thirdly, it might also be that your blood supply is not bringing enough oxygen around. So there's a diffusion, diffusion problem first. Diffusion is the movement from gas from your air cavities in your lungs, the alveoli, to the bloodstream. And that's a really thin cell membrane where the oxygen flows through. And if that cell membrane is damaged, or your lung tissue is damaged, or your alveoli are filled with fluid, then that gas can't diffuse that easily from the gas environment into your blood, and hence there's not enough oxygen getting into your blood. And lastly is a blocked circulation, and that can be either a physical block that your arteries are not able to bring blood to a certain tissue, or it can be that, for instance, your red blood cells are blocked by carbon monoxide and hence can't transport enough oxygen to your tissues. So a next question, I promised some interactivity. So what is the minimum level of oxygen needed to sustain life? And you can all put your answers in the chat window. Come on, people. Let's make it interactive. So you can answer the question in the chat window. Okay, so we've got a couple of questions, a couple of answers, and there's not one answer possible. So it was a little bit of a trick question, and let me explain that. So in normal air, there's 20% oxygen, 20.9% oxygen, and our body is optimized for that amount of oxygen. So hence our normal body function is able to function completely optimally at that 20% level. That means we can do full exercise, we can and still have enough oxygen in order to do what we want to do. So if we are sitting still and doing nothing, we can cope with less oxygen we can cope with less oxygen being breathed in. So, for instance, studies done with 10% oxygen, as long as you're sitting still, you're still awake, you're still conscious. It does mean your brain capacity is affected. So, for instance, one of the studies done in the University of Auckland in the Department of Exercise Physiology, they use a driving simulator. And if you're breathing 10% oxygen while doing a driving test on the computer, you will see that you make more mistakes and that you are less aware of what's happening around you. Although there are persons that 10% is actually not enough and they can't cope with it and they have to stop that study. And that's also determining on how optimized your physiology is. If you're young and fit and not a smoker, you can cope with less amount of oxygen than you can if your physiology is less optimized. If you're smoking, to answer your question, Tim, you are basically inhaling a bit of carbon monoxide. So that is making sure that a couple of your red blood cells or percentage of your red blood cells are not able to transport oxygen. And secondly, especially long-term effects of smoking cause your lungs to be not that effective in moving out all the pollutants that get into your lungs, and hence they get stuck there, and the tar will also create a blockage of parts of your lungs, and hence your diffusion also decreases in long-term smokers. So this was a experiment that they did in a hypobaric chamber, so a chamber where they can simulate altitude. And they went to an altitude where the oxygen level was really low, and you could see all the signs and symptoms of hypoxia. So we could see a shortness of breath, we could see aphoria, headache, not so much we could see it, but probably the person will have that afterwards. But you could definitely see from the outside a loss of concentration, hyperventilation, and they just intervened to make sure that he didn't get unconscious, but it was really, really close. And you can imagine if you would have the same effect underwater, where you become unaware of your environment. And as he explained, he thought that everything was well and he was performing really awesome. And the same can happen underwater if, for instance, our rebreather is not performing well and is not giving us the amount of oxygen that we need. So let's move to the other end of the spectrum, hyperoxia. So oxygen creates not only the O2 molecule, but also a couple of oxygen radicals, which are highly, well, I wouldn't call them dangerous, but they are highly reactive to all kinds of molecules that it can find on its way, including our protein, cell membranes, and our DNA. So when it comes into contact with one of these, it can actually damage it. If we increase, so in normal environment, those oxygen radicals are already there, but we as humans have built certain scavenger molecules, proteins, that can scavenge those oxygen radicals and make them neutral again. But if we increase the amount of oxygen we're breathing, we're slowly depleting these oxygen radicals, and at some point they can't keep up with the amount of oxygen radicals that are there, and hence they can start creating all kinds of damage. So If we breathe oxygen under hyperbaric environments, the partial pressure of oxygen increases further, or if we are breathing a mixture, and if we go deep enough, the partial pressure of oxygen increases as well. So at some point, we'll create a mixture that is not suitable for the depth we are. And there's also a time factor, which I will come to later. So hyperoxia or oxygen toxicity can happen if we're using the wrong gas mixture at the wrong depth for too long of a time. That wrong gas mixture can happen either deliberately by picking the wrong one, or not accurately mixing the gas, or analyzing the gas, or not marking the tanks properly. And hence, in that way, mistakes can happen, and you might end up breathing the wrong gas at the wrong part of your dive. And especially in technical diving, where we use various gas mixtures, including nitrox mixtures with increased oxygen percentage like 50, 80, or 100%, that might become a problem if you breathe those gases at the wrong depth. There are also a couple of drugs known that might interact and might lower our threshold for getting oxygen toxicity. And the most important one that we are using in diving is, or some divers are using it, is pseudoephedrine, which is a anti-motion sickness medication most commonly used in stickers that you can put behind your ear. Another known factor that interacts with oxygen toxicity is hypercapnia. If we have an increased amount of CO2 in our body, and we'll discuss that later on, we'll have an increased risk of oxygen toxicity. Lastly, in the hyperbaric chamber, when we treat patients either for wound treatment or for diving emergencies, we use 100% oxygen as well, and that can be at a depth of 15 to even 18 or 20 meters, depending on the treatment table. So that is really high oxygen partial pressures that we commonly don't use in diving. And the main difference is that in a hyperbaric chamber, you're sitting still, you're in a warm, comfortable environment, so you don't have that much increased levels of CO2. But more importantly is that if you would get an oxygen seizure in a hyperbaric chamber, we just switch you to air and you come around and we'll continue the treatment, or if it's a wound treatment case, we'll bring you back to the surface, and that's the end of the treatment for that day. Where in diving, if you get an oxygen seizure, you are most likely to drown. So hence, it's really important to choose the level of maximum partial pressure of oxygen relatively conservatively. And the question is, does the DNA in the cell get completely destroyed, cell die, or does it target a certain gene region? As far as I understand, but I'm not an expert in this, is that it just randomly attacks parts of the randomly attacks the DNA. So it might get a little bit damaged, and dependent on the amount of damage, the body might be able to repair it, or if it's too much damage, then the cell will die, and then it has to be replaced if possible. So the next slide is, I talked already a little bit about those limits, and for those of you who've done a nitrox or a technical diving course, you might remember some limits that you've been told. So what limits should we adhere to in diving as a maximum partial pressure of oxygen? And to answer your second question, MA, for proteins, yes, that's the case as well. It's randomly where it attacks the protein, and again, how severely affected that protein is if it needs to be replaced. So a couple more answers, and then we'll start the discussion about what maximum partial pressures to adhere to. Okay, so let's discuss the answers that I've seen mostly are 1.4 and 1.6 as maximums that are adhered to. And let's put that in a bit of perspective. So, so what I've explained already before, in the hyperbaric chamber, we use 2.5 to 2.8 atmospheres, but that's in the safe environment of a hyperbaric chamber. For instance, military divers that actually go underwater will most commonly use 1.7, or on an exceptional exposure, if they need to go under a ship to get to the other side of a military ship, then they're even allowed to go to 10 meters on pure oxygen, and hence have a partial pressure of 2. This is with a full face mask, so their chance of drowning is a little bit less, and they are well trained and super fit guys that are diving those oxygen rebreathers. So for us normal mortal persons, being a bit more conservative is a wise choice, and that's why most recreational and scientific diving is all based on the maximum of 1.6, which comes from the NOAA, the National Oceanic and Atmospheric Association. They made a table a long time ago that got basically copied into the rest of the diving industry, and especially in the 90s when the nitrox diving courses took off, the 1.6 got used, but because there was a big fight between the recreational settled diving organizations and a couple of new, more technical related diving organizations that were advocating for using nitrox, which in the beginning 90s was called a voodoo gas. They had hired an even safer partial pressure of 1.4. And there are even organizations like GUE nowadays that even uses lower partial pressures of oxygen, 1.3 for their standard gases, and for rebreather diving 1.2, where other rebreather set points are most commonly 1.3 because of their length of exposure. So it really, as I mentioned before, depends on the combination of that partial pressure of oxygen and time. So this is the exposure limits. Oh, sorry, this is not the right table that I put in. Sorry, no. So the partial pressure is the exposure duration is limited to. It's a combination of exposure duration and partial pressure. But already from half an atmosphere, 0.5 partial pressure of oxygen, there is a effect on the lungs. It's a slow one, and it slowly builds over time. It's what we call the pulmonary oxygen toxicity. And with continued diving, especially if you're doing technical diving with long decompressions using high partial pressures of oxygen, you might get to those limits where you might see a effect on the lungs, where you basically get a sort kind of inflammation, a burning effect on the lung tissue, which makes the diffusion of gases in and out of the lungs a little bit less effective. Luckily, that is an effect that will heal over time. But with higher partial pressures, somewhere between, above 1.6, we can get neurological symptoms. On really, really rare occasions, we might see them with slightly lower partial pressures, 1.3, 1.4, with long exposures or in combination with hypercapnia. So on the next slide, and I will introduce it before I put it on, I have a short video of a oxygen convulsion study done by the Americans, where they deliberately give a person high partial pressure of oxygen in a hyperbaric chamber.
This 1940s film from the US Navy shows the effects of breathing oxygen under pressure. As more oxygen is forced into the bloodstream, it quickly becomes toxic. The effects are debilitating and incredibly painful, and eventually, though not in this case, fatal. So at depth, where the gas has to be under pressure, the only way to stop the oxygen becoming toxic is to reduce the amount of it we breathe.
So the main symptom we saw here is the convulsion, but for those nitrox divers in the group, there are a couple of others that you might come notice of in the course materials, most often combined in an acronym, CONVENTED, where we have some visual disturbances, ear ringing, nausea, tingling, and twitching of the muscles, especially around the face and the lips. And some divers do notice these, and in that way are able to switch early back to a gas mixture with a lower partial pressure of oxygen, although that there might still be a case that because it takes a little bit of time for the partial pressure of oxygen to drop in your brain, that you might still get the convulsion. Other symptoms: irritability, euphoria, anxiety, and dizziness and dyspnea. So this is the table I showed you briefly, but this is not the central nervous system oxygen toxicity table, but the oxygen toxicity for pulmonary oxygen toxicity. And that answers your question, Tim, regarding the amount you can dive within 24 hours. In that way, you can keep track of your pulmonary oxygen toxicity by multiplying the, or taking the fraction of your dive time at a certain partial pressure of oxygen, divided by the maximum exposure, either per dive and per 24 hours, and hence by adding all those sections in your dive, especially in decompression dives, you can calculate the pulmonary oxygen toxicity dose. This is most often done in either decompression modeling programs or your dive computer as well. The so-called pulmonary oxygen toxicities, or OTUs. The other thing that we calculate is the CNS oxygen toxicity, and most organizations also calculate that in a percentage, similar calculation method with a table as well, although it has never been scientifically proven that the CNS clock, as it's been called, is actually accurate to track CNS oxygen toxicity. And what we know from technical divers that are doing extreme dives with long oxygen decompressions, where they get CNS oxygen percentages well over 100, even into the couple of hundreds, and still don't get an oxygen seizure or oxygen CNS problems, is mostly due to that the CNS clock doesn't capture what we do in those dives, where we what we call air brakes, or brakes to a lower oxygen partial pressure, and hence we can kind of reset that clock better. So to answer your question, Ryan, how does pulmonary oxygen toxicity compare to CNS? The oxygen partial, the exposure times for the table you see here are for pulmonary oxygen toxicity are far longer than the CNS table. So the exposure durations are far shorter for CNS. So that's why in most nitrox courses, you will only hear and see about the CNS oxygen toxicity table, because they are considered leading in normal recreational diving. When you start doing technical diving, you might come across this table from the NOAA. And if you're in a hyperbaric chamber and getting treated with a treatment table, we also calculate the pulmonary oxygen toxicity dose, because that does become a problem when you're doing repeated treatments in the hyperbaric chamber. So the last gas I would like to discuss with you guys is carbon dioxide. So carbon dioxide shouldn't be in your inhaled gas. Although if we have a faulty rebreather, for instance, the scrubber is not working, you might get inhaled CO2. Or if the compressor is not well maintained, or the compressor is sucking in air from the environment where there are combustion gases, which can also have CO2 in them, or if the compressor is running too hot, it can produce CO2 as well. But more commonly, we get CO2 by actually the production of it by our own body. And normally we would breathe that out when we exhale. So when we are not capable of getting rid of our CO2, then we can get into what we call hypercapnia. So how can we get there? First of all, if we are using a gas mixture like air at a depth, then at an increased pressure, then the gas density increases because there are more molecules per volume. And at a certain point, you can, with an overexerted example, it's like breathing a liquid getting a liquid through a thin straw. We don't experience it that much, but we do need to take more effort in order to get the air, the hyperbaric air into our lungs. And for instance, at 30 meters, not a really exotic deep depth, we are already experiencing that our maximum exercise capacity that is limited by ventilation is already halved. So that means at 30 meters, you can only do half the amount of the maximum exercise that we're doing. Secondly, exercise also increases our CO2 production, and hence the combination causes that we at some point can't ventilate enough to get rid of the CO2, and hence we can get an increase in CO2 in our body. Stress causes an increase in hypercapnia as well, because your muscles are working harder, but also stress can be induced by hypercapnia, and in that way we can get into that spiral again that can go downwards if we don't intervene early enough. So your question, if that exercise capacity is limited by, if it's dictated by oxygen partial pressure, no, it's irrelevant of oxygen partial pressure. And even with air or a nitrox mixture at 30 meters, your partial pressure of oxygen is way higher than your oxygen partial pressure at the surface. So oxygen is not the limiting factor to get it in. It's just the amount of breathing just takes longer because it's a thick gas you're breathing in and out, and hence it takes more time to get it in and out. And the other problem is, and then we're getting a little bit technical, so your excuse if you're not getting this, but if the gas flow is high, we get a turbulent flow, and turbulent flow moves easier through our airways, where if the gas density increases, at some point you get laminar flow, and laminar flow travels far slower through our airways. So all in all, by increasing the gas density, we increasing the work of breathing, and hence our exercise level is reduced because of CO2 buildup. Another way to build up your CO2 in your body is extending your dead space. So the top part of your airways are not part of your gas diffusion. So every time you exhale, the lowest part of your lungs, where your highest amount of CO2 is, gets to the upper airways, and then with the next inhalation that moves down again. So there's a little bit of inefficiency there. But we're able to cope with the amount of dead space that we're having. And we can extend that a little bit, for instance, with a small snorkel. But if we increase the diameter of that snorkel, we're increasing dead space, and hence at a certain point we can't get rid of enough CO2. And the same applies to skip breathing. If you're hypoventilating, not breathing enough, you don't give your body the opportunity to get rid of CO2, and hence you get hypercapnia. So the symptoms of hypercapnia are similar to hypoxia. Fatigue, headache, sweating, hyperventilation, and at some point anxiety and panic, and that's that downward spiral that I talked about. And at a certain point, if the CO2 gets high enough, you get unconscious. And again, there are tables out there that show that there is a short period, a couple of minutes of a increased amount of CO2 is okay. But if you get more, if the duration is longer, you can endure less CO2. So how can we prevent hypoxia, hyperoxia, gas narcosis, and hypercapnia? And the main way of doing that is selecting the right gas for the right dive. So especially when you're diving deeper, and in my case, I choose that limit to be somewhere between 30 and 40 meters, depending on the environment, that there needs to be an increased amount of helium, because helium is a non-narcotic gas. It's really light, so your gas density goes down, and in that way, we can get rid of the inert gas narcosis problem and the hypercapnia problem. So let me explain what I mean with depending on the environment. So for instance, a really nice reef dive without any currents in really good conditions, I would consider diving air to 40 meters. But if it's a challenging dive in a dark environment, you have to work hard, and you might be even tasked to do certain challenging tasks, to do certain things during a dive, like taking pictures or taking measurements. I would limit air and or nitrox to be to 30 meters, or even consider using adding helium to that mixture in order to make it easier to work at that depth. And that's also dependent on your personal level of comfort with narcosis. So, and lastly, oxygen has to be chosen as well in a relative way, that based on our maximum depth, our maximum pressure, we need to choose the oxygen partial pressure not to exceed the maximum, and even and be mindful that if we start doing really deep dives, we need to make sure that we don't breathe that gas mixture at the surface, because the partial pressure at oxygen might be too low to sustain life on the surface, especially walking on the deck or being busy in the water at the surface. Those are the dangerous moments. It's inert gas other than yeah, so helium is also an inert gas, mostly. There are some studies out there that show that helium has some neuroprotective effect, but mostly we would consider it to be an inert gas. So inert gas narcosis is something we call, because there are a couple of gases, like for instance oxygen can be narcotic. We're not entirely sure. There are studies showing that oxygen is narcotic, similar to the amount of nitrogen, and other studies show that oxygen is not narcotic, so we're not actually sure if oxygen is narcotic. So yes, it becomes the term inert gas narcosis. It might not be the best term that is used in the diving industry. It might be just gas narcosis, gas-induced narcosis. Any other questions? I will open the floor and I will unmute you all, so you can.
I've got the big C, so I need some finances.
So you can choose to either answer,
wow.
take your question by either by typing or just shout them out, and you can put on your video if you want as well. Oh, sorry. That one.
I think so, yeah.
Any questions regarding any of the gases?
The background and all his bubbles.
Yeah, it's bubbles, yeah.
There's a right there, and like a masala has the background. I was asking, like this, you remember masala went to the chamber. Was asking, when are you going to? We have something about the chamber.
So Ryan, your question regarding what we call isobaric counter diffusion. It's a interesting topic, and definitely, well, slightly related to this presentation, although I didn't cover it because it's more decompression related. So there's a theoretical thing called isobaric counter diffusion, and the experts are not sure if it actually exists. So there are a couple of interesting papers out there, and if you're interested in reading more about that topic, please let me know, and I can send you I think the latest paper by Simon Mitchell is the most interesting one to read about that. So if you can either send me a text message or a text message, that would be the easiest way for me to be reminded to send you that paper. Okay. Yes, it's something I figured out as well. I saw it by others using in Zoom. So if you in your video settings, if you take your video button at the bottom saying stop video and then the arrow on the side, you go to video settings, or sorry, you can choose there directly your virtual background, and that's where you can select the background, and the image in the background is just any image you can choose to put in there.
Okay.
So I have to do this.
Yeah.
So, but you can't find it.
I'm not going to do that.
Yeah, so the GABA receptor is something that is most anesthetics actually work on that receptor, and well, by incidence the nitrogen works on that as well. So the funny part is that the fields of anesthesia and gas narcosis research in diving medicine are actually quite related.
Oh, then you could add an image. Oh, I see, right.
So put an image then.
Oh, I don't know. I'm not going to start.
Okay, are there any other questions?
I did. That was, how do you make it? You did it by doing this virtual.
But I asked about his project. No,
oh, what was the question?
Yes, I was talking about like a Marcella. As I said that Marcella is
Quickly.
Here. Marcella is here. She participated on
yes,
the
okay. Keep going.
Here. And then I said, like, are you going to publish anything about the results or whatever?
So the question of Marcella, yes, I'm intending to publish my results of my study. Actually, my first publication just got published today in the, oh no, sorry, is recently accepted and will be published in the next journal of diving and hyperbaric medicine in June. Sorry, so that's the first one. And soon after I hope to publish the other results as well. So yes, we're in the process of writing and getting our results published, but the analysis takes a long time, and then writing it up and agreeing with all the authors on what the manuscript looked like. So it's a long process, but it's definitely coming.
Thanks. Sorry, I hadn't realised I'd unmuted the microphone. Never mind, that's okay.
No worries.
Xavier, can you share that paper with us? Because I'm not sure if you can get that through University of Auckland, but I'm pretty sure because Canterbury doesn't even have a med school, I won't be able to get it down here.
Yeah, I can definitely share that paper. Just send me a text message in either WhatsApp or Facebook Messenger, and I can definitely share that paper with you.
Thanks.
So
Marcelo would like that as well, if possible.
yeah, please send me a message. That's the easiest way to not forgetting about it.
Yeah. Okay.
So Tim Diego was asking, so are the effects of nitrous oxide different to nitrogen narcosis? So nitrous oxide works on the NMDA receptor, where nitrogen works on the GABA receptor. So their pathways are slightly different, and hence their symptoms are slightly different, but in the end, they both impair your cognitive function. Yeah. Cool. Are there any? Yep. Yep. So there are a couple of other gaseous anesthetics and also liquid anesthetics like propofol that are acting on the same GABA receptor. Although the biggest difference is that they are far more narcotic than nitrogen and nitrous oxide.
I've got a quick question.
Yeah.
So basically, under pressure, obviously you would expect, I guess the best would be sort of like a contraction of cells and that. I mean, our bodies aren't meant to be going, you know, like 30 meters deep, 40 meters deep. Something would be happening. Do you think the cells are actually changing in regards to, could the receptor sites be changing, and that's why we're actually getting the symptoms instead of sort of gas being necessarily. I mean, obviously the gas does cause problems, but at a certain pressure, your cells would change slightly. Or am I?
Sorry, do you mean the salts like the sodium, potassium, and
Oh
chloride?
no, it's your cells. So.
Oh, the cells. Sorry, what are? Then I didn't understand your question.
Because at pressure, you expect the structure of things to change slightly, correct?
Well, the GABA for nitrogen, the GABA receptor is blocked, and hence its signal pathway is blocked. So the neurons can't talk that effectively to each other anymore.
Yeah. So is it blocked because the receptor structure is changed slightly? So I don't know, how much do you know about biochemistry?
A little bit. So the GABA receptor has, so there are variations of the GABA receptor, and certain types of the GABA receptor probably have a hydrophobic spot where the nitrogen molecule fits in and hence blocks its way of transforming to an open and closed state.
Okay, yeah, that's answered my question. I was interested in how it's actually affecting it at a certain level, because obviously you could get that maybe it's not even fitting anymore. I mean, in some cases.
Yeah.
So, okay, but that answers my question. Thanks.
Okay. Any? Interesting question. So hydrogen is slightly narcotic as well. I'm not entirely sure if that the fact that it is diatomic would cause it to be narcotic. The most important part is how it can fit in that hydrophobic spot of the GABA receptor is due to the molecule being hydrophobic. So that's where, when I talked in the beginning, the solubility in olive oil is actually still relevant, although there are limitations to that. Because, for instance, there are certain really big molecules that are still really hydrophobic, but at some point there seems to be a cutoff where it no longer fits into that hydrophobic spot of the GABA receptor, and hence it's not narcotic anymore. And there are a couple of other molecules that, based on their hydrophobicity, we would expect them to be narcotic, but for some reason they don't fit into that spot in the receptor, and hence are not narcotic. And there are a couple of molecules, slightly bigger molecules, that have a left and a right orientation, and one of them seems to be narcotic while the other is not. So it is quite a field, a specialty on its own. Okay, I think we, unless there are still other questions, I think we can end this presentation. I would like to thank you all for joining this presentation, and I hope to see you again in one of the next AUUC webinars that we are going to organize. So see you next time.
Thank you for an excellent presentation.
Thanks.